It was rather a different message because functional amyloid is produced by bacteria and other organisms to do something good, unlike the disease-related amyloidosis proteins. And my message was that when nature wants to do something well, she does it in a very controlled and organized fashion. So the interesting thing about bacterial amyloid and amyloid made by humans as well, for that matter, is that everything is under control by the cell, which means in practice that you have not just one protein aggregating, but a lot of proteins that help the protein go to the right place and only to aggregate when the time is right. And the other aspect is that the proteins that do aggregate have been designed to do so. And in bacterial amyloid, we've learned that the really good amyloid is produced by making proteins that contain a number of imperfect repeats. So it's basically copies of a short sequence that are hitched together in a protein. And each repeat is able to form a small kind of amyloid. So if you combine them all, they become very good at forming amyloid, which goes directly to the amyloid state and doesn't go about making toxic intermediates. Also, the aggregation, as we've also learned, is very controlled and slow, so it doesn't form explosive growth increases, which is difficult to control, but it happens in a very steady manner where you just have the protein forming a nucleus, which then slowly extends. And we've uncovered the basis for that and shows that that contrasts with pathological amyloid. So that's an important message showing how nature really is good at making potentially toxic stuff.